and transport fluids. In the respiratory system in particular, they usually transport mucus, and the mucus is what captures all the inhaled debris, the pathogens, the pollutants we inhale all the time. So I think I've read our internal surface of the lung is like a swimming pool. So you can imagine how much opportunity there is for dust to settle. And it would if there wasn't this continuous clearance operation going on at all times. And to give you an idea what that looks like in real time, this is something we can actually visualize, record with high-speed technology. That's really our focus, to understand these mechanics. Now, normally, as I said, we don't even notice this process. It's just there and helps us breathe. But when it does fail, when it is impaired, it actually is associated with a lot of very severe, chronic, and mostly still incurable diseases. Let me see. This is, yeah, including chronic obstructive pulmonary disease, asthma, cystic fibrosis, primary celiac dyskinesia, fibrosis, you name it. Almost all chronic lung diseases also have either an underlying or a even driving a mucociliary clearance defect, affecting almost 1 billion people worldwide. And there isn't really any cure. We don't understand how to fix it. Like, once these cilia stop moving, basically, we don't know how to turn it back on. One of the issues to sort of restore that, towards restoring that, rescuing that, is that we actually, for the longest time, didn't even have a goal, a target post. Like, what is actually effective clearance? So if I go back to this video, I don't know if this is possible. Let's see. Yeah. I mean, can you tell me, is this good? Is this bad? I mean, they're moving, but is this good? We don't know. And this is how, over the longest time, the field has looked at it. It's just qualitative understanding. Okay, it's moving, I guess. And then maybe some experts would be able to say, well, this beat pattern is not normal. But there aren't like many quantitative metrics on this that makes it really hard to develop disease models and actually working on restoring it. So the first thing that I did when I started my team was to actually look into this problem. And you all know about all these atlases, lung atlas, so forth. We kind of worked on a mechanical atlas of the respiratory system. So we were able to access healthy human lungs, also animal lungs for comparison, and dissected all the different branches of this organ into tiny tubes that we can then open to visualize the active ciliary movement. This is, real human ciliary beat on those surfaces, and the associated clearance of fluids and particles, and relate that to the underlying cellular structure. Now, what I've realized when I was listening also to Dominic's talk, I should have talked about the step that I'm omitting here, but at least he gave a little introduction into physics or biophysics-informed modeling. So what we do with this information is not just simply have nice videos, but we extract a lot of quantitative metrics from this, and this is, not AI-based or anything. It's image processing, signal processing based, where we extract, for example, the amplitude of the beat, the frequency, the distribution of the beat, and we also measure all the flow dynamics that result from that. And we connect these things mechanistically, not by correlation, but by physics-based models, bottom-up constitutive modeling, numerical simulations of the forces put into the fluid by the ciliary bead, resulting in fluid transport. Which means, with these data from these healthy lungs and the animal models and some disease studies, we were actually able to construct something like a human reference benchmarks, mechanical benchmarks in this sense, which give us, a way to measure distances between tissue states, to say this is more disease than another state, and we're further away from health than this one, and also assess our in vitro models to say this is a good model of healthy clearance, and this one isn't. So what I want to say is we can now say, if you tell me how many cilia there are and what's their beat frequency, I can tell you sort of, is this good or is this bad? But I can still not tell you how to change it. So that is where we're now. We're basically at the point where we can understand kind of starting here all the way up where the problems lie. If there is a problem, we can identify this purely by physics and mechanics. I mean, at least on the level of mucociliary clearance. But what we still don't know is how is all of this regulated? How can we actually change it, right? We're still dealing with sort of a black box. And this is where this new collaboration comes in. We want to understand what are the molecular processes, the signaling processes that actually change those individual parameters that we can already measure and that we can use to predict function. And so on the one hand, this will allow us potentially to go backwards and say, once we have a certain phenotype, we can maybe say what it means for the regulation on the molecular level, but also reversely, once we have these omics data sets, maybe we can actually predict what the clearance function is eventually, which would be amazing. So to start on this, from a very sort of simple starting point, we teamed up with Fabian Theis' team to conduct a big perturbation study. And in this case, we basically take some advanced human airway models, including organoids, liquid interface cultures, and now starting also organ chips. And we use compounds and factors that we know impact clearance. But no one really knows why and how, but we know they change the phenotype. And we're combining right now several data sets, the phenotypic clearance data sets, immunofluorescence data sets, and single-cell RNA sequencing data to see if we can sort of reveal this trajectory of the relationship between these different scales. And the idea is that with a model that the Theis group develops called CellFlow, that we can not only understand the regulatory mechanisms underlying mucociliary clearance and sort of evaluate further this distance between healthy and diseased states, but also predict what the impact would be of novel perturbations, combinations of factors or drugs that we haven't tested, to modify clearance and maybe build better disease models, build better healthy models, but also eventually find some promising drug targets. Now, okay, that was the theory. Then in practice, the first thing we had to figure out, I just want to show you sort of the journey, was to figure out how to actually do single-cell sequencing on our models, because the standard workflows offered, or the protocols offered by the suppliers just didn't work. And I want to say, we're a biomechanics biophysics lab. We usually get stressed out when we have to do one qPCR. So going to this was a big step. But with the hard work of my team, they actually figured out a new protocol that especially works for those airway cultures, which has just, if you're interested in this, of course, I'll be happy to explain. But basically, instead of fixing the tissue and dissociating later, the trick was to dissociate first and then fix, and apparently that solved all the issues, and we got nice cell counts, high cell viability, and good quality RNA for the further processing. So with that, we did some first validation. We basically compared the cell types that we can identify using this immunofluorescence staining. That's the major cell types and the rare cell types in our cultures. And looking at the healthy conditions or control conditions, we can verify that these cell types are still present in our single-cell data, which sort of gives us confidence that we're not losing some rare cell types on the way. So with that, we now, of course, still have the problem, how do we link this to our clearance function? Because I showed you earlier that we're studying clearance functions, but those are quite work-intense imaging procedures, not really high throughput to identify all those mechanical parameters that underlie clearance. So we had to come up with another way to quantify good and bad clearance. So this is an example from looking at one of these model cultures, where you see a sample that has good clearance, so basically really moving the mucus and the trapped particles that are fluorescent around and away from the tissue, whereas a case of bad clearance where the mucus just kind of gets stuck with all the dirt in it. And we found a way to quantify this using just snapshots of where the mucus and where the particles are over time. And this allows us to scale this into 96-well plates and further, and basically assigning good clearance, bad clearance, and intermediate clearance by eye, but of course we don't want to do this by eye, but we developed an algorithm that basically uses these images to extract spatial and frequency space parameters and gives different scores to each clearance phenotype. So then we can right now have a very crude classification. We want to improve this, but right now it's really just like good, bad, or in the middle. But at least now we can map each single cell RNA sequencing phenotype, so to say, to a clearance phenotype. In addition to that, we have other measures that we can get from these samples: ciliary bead frequency, speed of the flow, and staining information. And so with this, to summarize, what we've been focusing right now is just really getting a protocol working to do single cell sequencing, making sure this works, and also building a way to generate a distance matrix to the atlas, human airway atlas. And the next step now is to integrate all these different data types that I showed to actually get to the point where we can see, is the model that we're using actually predictive? Can we actually use this? Can we already bridge the gap between molecular information and actual tissue scale clearance? And with that, I would like to thank my team and my collaborators and my funding sources. I'll be happy to take any questions. Okay.
Thanks, Jana. Do we have questions?
Everything clear?
So I have one question. I would like to understand how far can you go with modeling, like basically this the human lung in a petri dish?
Mm-hmm. You mean how close is it?
Yeah, I mean how realistic, what is missing to understand?
I mean, it depends on what you want to model, but this particular aspect, barrier and clearance function, it is very close. I mean, we get very similar clearance effectiveness, you could say. Of course, once you want to study really immune responses or muscle responses, you have to add other cell types in there, the smooth muscle or immune cells, and make the model more complex. So this is really just focusing on the surface.
Okay, yeah.
So that really depends on what you're asking. But for the clearance aspect, for the mechanical clearance aspect, I think we're really close.
Okay.
Yeah. Okay.
Do we have another question?
No.
And about the CellFlow model, like I kind of understand that the perturbations would also be potential treatments and so on.
What?
The CellFlow model?
Yes.
I'm wondering how much you can really predict like out of distribution perturbations, or if it's only, you mentioned it's working for like new combinations of perturbations?
Yes, so the idea is that you test, I mean, there's a publication on that, you know, from the Theis lab, but basically the idea is you don't test all possible combinations of these factors that you have. You test a subset. Mm-hmm. But then the subset is enough to basically, or should be enough to train the model to predict the outcome of new combinations.
Okay, but it's only combinations, not like if you have a new
It's not
factor
yet a new, completely new factor. It's not at that point, but maybe if we know enough about the new factor, if it's interacting with similar pathways, one can make a prediction. But right now, the focus is really like only can we predict new combinations? This could be, for example, if we have a risk factor for asthma, and now we combine it with a preventative factor. Yep. You know, like this also. It's like something that would maybe happen in the environment. Like you have a certain exposure to an allergen, but at the same time you inhale the cow dust that we heard about. So is this going to be enough?
Okay, thanks. Okay. Let's thank Sarah again.
Thank you.